start. Let me see. Very good. So thank you very much, everybody, for being here in this second seminar on the seminar series of complex systems organized by the area of complex systems at the UAM Iztapalapa. Today we have the great pleasure to have with us Andrea De Martino. So I'm going to briefly introduce him. Andrea did his PhD in a CISA in Trieste under the supervision of Professor Matteo Marsili. If I recall correctly, it was in the area of agent-based models in economics, in particular minority games and variations of the minority games. After that, he did a postdoc with David Gross in Germany, studying thermodynamics of rotating stars, if I recall correctly. Then he landed at the Parisi group at the University of Rome La Sapienza, and he stayed there for a number of years. Currently, he's at the Italian Institute for Genomic Medicine in Torino, and he just announced that he's going to have, he just got a new position, okay, at the Politecnico di Torino. So, Andrea, thank you very much for being here. It is a great pleasure to have you with us. I know that you wanted to practice a bit your Spanish, so I'll let you to say some words in Spanish, and then we proceed with the seminar. Thank you very much. Go ahead.
Bueno, quiero agradecer a los organizadores para invitarme. Es un verdadero honor para mí estar aquí, pero yo quería hablar español solo con Isaac porque soy un principiante, ¿no? Can I share my screen? Should I start?
Yes, please, go ahead.
Yeah, okay, let me see. Wow. Oops. Okay. Can you see my slide, my white slide?
Yes, we can.
Okay, great. So thanks again for inviting me. This is actually the title of my talk. Let me just minimize this. Okay. I'm in Torino, as Isaac told you, and my physical location is actually there. It's the blue spot. We're in the northwest of Italy. We're close to the border with France. That's where I'm speaking from. So my talk is about the growth rate of bacteria. So this is not going to be very technical talk. I think there's going to be some technicalities, but I just hope I will be able to give you the idea about some new stuff that is happening at the interface between physics and biology that I personally find very exciting. And basically, it all concerns the movie you're watching. I downloaded from YouTube. So this is just a bunch of E. coli bacteria that are planted in a suitable medium. I guess this is a glucose-based medium, and they're after some time that it takes them to adapt to the medium, they start replicating, and the population grows and grows and grows, and the number of bacteria increases and increases and increases. I think this movie is, the speed of this movie is actually increased a lot compared to the way it actually works in reality. They're not so fast in replicating, otherwise we would be in huge trouble. But anyway, from a mathematical viewpoint, the way you describe this kind of dynamics is just you say that the rate of change of the number of bacteria in the population is actually proportional to the number of bacteria, and the proportionality constant is what you call the growth rate. So this is just an exponential growth, and the rate, which is called the specific growth rate in the literature usually, I will mostly denote by the letter nu there, and N0 is just the initial number of bacteria that you planted in your dish. Right, when people started looking at this kind of systems, what they made was essentially extract from this data what people call the growth curves. So the growth curves is just a picture, a plot of the logarithm of the number of bacteria normalized by the size of the inoculum, so divided by N0 as a function of time. And this kind of sigmoidal shape that you see here is something that actually, let me stop this, otherwise I'll go crazy. So it's something that actually you see for many different cell types. It's not actually universal, even though many people think that actually sigmoidal curves is what it should be like. That's not actually true. But for many different cell types, you see this kind of sigmoidal behavior where you have an initial stasis period that can last for a certain time, which is usually called the lag time, before an exponentially growing phase of exponential growth. And here you start seeing the mess that we get into when we start looking at papers. Sometimes they call it lambda, sometimes they call it mu. The specific growth rate is just the slope of this line here. And then at some time, the population stabilizes and enters a phase that is called the stationary phase. And this is just summarized here. So the lag phase, the growth phase, and the stationary phase. This is sort of the usual picture of growth curves in bacterial or microbial or cellular populations that are growing exponentially. Now, this black line here is just one of the many mathematical functions that you can throw across the experimental points here, which are the green dots, to fit them. And there are many different sigmoidal functions that are used in the literature to fit the data. So there's actually a lot of ways that once you have the growth curve, you can throw a line through the points to describe them. But it's very, very hard to come up with a mathematical model that will give you this behavior. And this may be surprising, okay, but trust me, it's not easy. And the hard part to get it mathematically right is, of course, the initial phase, the lag phase. It's relatively easy to get the exponential phase, as you see here, and it's relatively easy to get the stationary phase, as you see here. But the lag phase is really, really hard to get mathematically. So even though these data are easy to fit, they're not easy to model. That's the message I want to give. On the other hand, what is easy to do is to basically describe how the specific growth rate, or the growth rate, I will mostly call it the growth rate, changes when you change the amount of substrate in the medium. So the stuff that these cells use and metabolize in order to grow. And this is just called S in this slide here. And S stands for substrate, which is essentially usually some sort of sugar like glucose, fructose, lactose, galactose, something like that. And as you increase the amount of glucose in the culture, so you make different cultures with different glucose levels, what you observe is that the specific growth rate increases with a sort of hyperbolic dependence on the substrate level that is usually represented by this mathematical function. And there is actually a very simple mathematical description here that works really well to describe data in what is called a Michaelis-Menten function in chemistry or in biology. Okay. All this is actually really, really, really old, and it was, as you can imagine, it was established decades ago. And this is the historic paper from which, the classical paper from which this plot down here was actually taken. It's a paper by Monod, 1949, The Growth of Bacterial Cultures. The title is really perfect for the contents of the paper. However, every now and then you really want to read a classical paper, right? You don't go back many years, but I actually decided once to go and read this paper from the 1949 Monod paper, because everybody cites it. And well, when you read the abstract, well, it says introduction, but it's basically the abstract of the paper. It says, the study of the growth of bacterial culture does not constitute a specialized subject or branch of research. It is the basic method of microbiology. So what it says here is that, look, there's nothing really to understand here. What I'm going to talk about is just how to grow bacteria on a dish. Okay, so this is not super encouraging if people want to understand something about these bacterial cultures. And as a matter of fact, after these papers was written, Monod didn't really work anymore on these things. He kind of shifted and started looking for the molecular mechanisms that determine different behavior of these types of cells in different media. But on the other hand, there are a bunch of other people that decided that it was actually worth to look more in detail at these cultures, especially to understand what today we would call system level properties. So what is the, in other sense, is there, the question that the people were asking is, can we understand something about the way and the key mechanisms, not in terms of individual molecules, but in terms of whole cell systems that allow bacteria or any other cell type to start growing? Okay, and if you think about it, this is a really, really deep question, because the state of growth, of cellular growth, is actually very non-trivial from a biological viewpoint. It's actually thinking about it, thinking about the amount of different things that have to happen in a cell in order for it to grow, you tend to believe that it's really a special, very, very special state of things for cells. So it's tempting to try and understand something about it. Now, one of the really important points in this story was the definition of a state, which is called the state of balanced growth, that could be used as a reference state. So if you come from physics, basically, I think a very good analogy of what happened in biology at this point was more or less thermodynamics. So in thermodynamics, we are used, because that's the way you do things, to study how systems respond to perturbations in a single parameter. When you change a single parameter of the systems and you keep all the other parameters fixed. And it really doesn't make sense to change two parameters together, because then you have to specify along which line you change these two parameters, okay? Now in biology, people were changing essentially all parameters at the same time. So results were really hard to interpret until they came up with the idea of balanced growth. So this was, I mean, it doesn't sound much to us, but it was kind of a revolution in biology. And the idea of balanced growth is simply to look at what systems do only when they are in the exponential phase. So instead of looking at the whole growth curve, you just focus on the exponential phase, on the phase in which bacteria grow exponentially, and imagine that it would just go on forever and ever. So you never reach a stationary phase, and you ask yourself what happens in that phase and in that phase only. So this means that you can essentially use the substrate level as the only, let's say, gauge that you tune in order to change the growth rate. Now, in practice, what happens in balanced growth, which is what you see here, is that as time goes on, during the exponential phase, all the quantities that we as physicists would call extensive, they grow exponentially. So this is what you see here. So essentially, things like cell size, which is shown here, or the number of cells, or I don't know, the overall population volume, they all grow exponentially in time. But intensive quantities, like ratios of extensive quantities, they stay constant. This includes, for instance, the concentrations of molecules inside cells. So this is a really useful state. And if you want to think about it in a microscopic way, you have to think about this, that when you see a population of cells that is growing, they are all different from each other. They're not identical, even if they have the same genome, which sometimes is the case, because you build your experiment in such a way that that's the case. They are all different in some specific, like they are different in the sizes at birth or at division. They're different in the timing of the cell cycle. They are different in molecular content. It's not like they have exactly the same number of each protein or of each RNA. So they are heterogeneous in this sense. But the great thing about balanced growth is that during balanced growth, each of these quantities, they obey stationary probability distributions that can be measured in the lab. And this is an example of what you find when you measure, for example, the interdivision time of a bunch of E. coli cells in different media, from very good ones, so media in which cells replicate very, very fast, to not very good ones in which they replicate very, very slow. Okay, and these distributions are actually stationary. They don't change over time. It doesn't matter where you perform your measurement along, as long as you are in the balanced growth regime, you will always find the same distribution. So this is really, really helpful. So what people have been doing is looking for, let's say, the equivalent of thermodynamic laws inside balanced growth regimes. And one of the first interesting thing that people have found, this was actually done by a group in Copenhagen and has become really, really famous for these works in the late 50s. They found something really, really specific, and I hope to be able to show you how special this is. Okay, so when you, what they did was essentially by studying different cultures that were growing at different specific rate, and here they call it lambda instead of mu, but it's basically the same mu as before. They measured the ratio between their RNA and protein content. So they just took the cells, they squashed them, they took out all the RNA, and they took out all the proteins. They are easily separated in the lab, and they just measured the overall mass of RNA, the overall mass of protein, and they did the ratio. Okay, it's something that could be done already in the 50s in the lab. Now, the thing about this ratio, RNA over protein, this is something that I won't show in detail, but you have to trust me about it. It's really easy, it's just time consuming. It's actually proportional to the fraction of the overall protein mass that is devoted to ribosomal proteins. Now, ribosomes, as you probably know, is the machinery that produces proteins. It's a complicated thing. It's made by many RNAs, many proteins. It's a large, complex molecular machine that is devoted to the production of proteins. So the amount, the overall fraction of ribosomal proteins over the whole, I mean, how many ribosomal proteins you have over the whole proteome is actually an interesting quantity to look at, and it's just proportional to the ratio of RNA to protein. And what they found is that as you change the growth rate, this quantity behaves in a highly, highly reproducible linear fashion. So basically, if you want to write down a mathematical representation of this behavior, you would have to write something as easy as this here. There is an intercept here that tells you that when the specific growth rate goes to 0, you still have a residual amount of ribosomes in the cell, and then it increases linearly with the growth rate as the growth rate goes. Now, I should say that this linear increase cannot go on forever. Of course, at some point, it has to saturate up here. And also, there are some small deviations down here, so it kind of tends to saturate down here, but it doesn't, I mean, this doesn't change the fact that you still have a non-zero intercept here. So maybe the linear function is not a perfect fit here, but it's actually a very good fit. And if you've ever looked at biological data, this is actually as good as they will ever get. It's really, really hard to find biological data that are so clean and so nice. So this is something really robust and reproducible. And the question is, where does this come from? So why do cells increase the amount of the fraction of ribosomes over the overall protein as the growth rate increases? And notice that this is something that occurs more or less universally. So people have tried to measure, ever since this 58 paper, they have measured it in a huge, over a huge spectrum of cells, from plants, mammalian cells, I mean, you name it. They always find roughly the same thing. So there's always some linear dependence. So this is a very robust result. Going on, they also looked more carefully at other types of proteins. So not only ribosomal proteins, but also, for example, what are called catabolic proteins. And if you don't know what that means, it's not really especially important. As long as you know that catabolic proteins are the proteins that are necessary in order to process sugars. So usually the substrate that is used for growth is some sort of sugar, and catabolic proteins are the ones that allow you to process these sugars in the metabolism. And they also found very highly reproducible linear behaviors of this type. In this case, the fraction of catabolic proteins tends to decrease as the growth rate increases. So essentially they started building up pictures that were telling you that these cells are doing, as they grow in the balanced growth phase, depending on how good the medium in which they are, so depending on how fast they can grow, they basically change the pi of the proteome according to what they need the most. So in this case, this pi represents 4 different types of proteins. You have housekeeping proteins that more or less you always have to synthesize. Then you have catabolic proteins, you have ribosomes here, and you have other enzymes, generic things. So what you find, what these plots tell you is that when you modulate the growth rate by changing the carbon availability, so for example, this is a condition that is usually called C limitation in the literature. What you find is that as the C limitation increases, which means the growth rate goes down, the fraction of the pie devoted to catabolic proteins increases, like you see here, while the fraction of the pie devoted to ribosomal proteins decreases, as you see here. Okay. Now, the interesting thing about this is that these fractions are always behave in a way that is proportional to the growth rate. And this is something really interesting if you think about it, because the fractions are essentially a ratio between the mass of the proteins that you're looking at and the overall proteome mass. And it's not really clear, I mean, it's not really easy to understand how the ratio of these two quantities can behave in a linear way. So this makes you think that with the growth rate. So this makes you think that there is some, you know, really crucial regulation going on here that gives rise to this robust behavior. And then, of course, the more you look, the more you find, and they found this similar linear behavior in other proteome sectors. For example, in respiration, you observe this kind of behavior. In fermentation, you observe this kind of behavior. These are all cellular functions that are important at different stages of the cell life, and they all behave in this way. So there is even if in principle there is a myriad of molecular mechanisms that are going on, and there's metabolism, and there's gene expression, and there's, you name it, thousands and thousands and thousands of processes that are random and stochastic. There's Brownian motion. There's a lot of things going on. At a statistical level, they give rise to sort of highly reproducible, robust laws. Okay, and there's more. And I will not, I mean, I could describe more, but basically the idea here is the following, that at some point there was a picture that was building up that evolutionarily distinct cell types, so from microbes to cancer cells, display highly similar physiological behavior when you look at them in the state of balanced growth. Okay, so when you do the biological equivalent of doing thermodynamics, you can identify something similar to the thermodynamic laws. So something robust, reproducible, that seems to be happening in any cell type you look at. So the kind of changes in the proteome organization that I've talked about so far are called growth laws, but there are many more of these, let's say, statistical regularities that have been found over time. And I will talk about a few of them in a while. So the question that, of course, immediately arises is, where do they come from? Okay, so why is it that these systems that are unrelated, practically unrelated from an evolutionary viewpoint, they perform very different tasks? You know, how come they behave in the same way? Now, you got to think that essentially the way in which the proteome of a cell is allocated, the different parts of the proteome are allocated, tell you something about the functions that these cells want to perform. So for example, if a cell starts allocating more proteins to ribosome, then it means that it's likely that these cells have to produce more proteins, okay? Otherwise, they wouldn't do it. Okay, so there was a simple answer to this question that was given, again, a long time ago, that has to do with selection. So cells, basically what they want to do is they want to replicate as fast as possible. So there is an intrinsic drive here to maximize the specific growth rate. And of course, this is really intuitive. So if you imagine a bunch of cells growing in a certain medium, right? And there is one cell that is growing faster than the others because maybe it's using the sugar of the medium more efficiently than the others, then it's just a matter of time. Eventually, it will outcompete all the others in using the sugar, and in the end, you will just find that cell that has the largest growth rate inside the population. So this is totally intuitive and makes a lot of sense. And it also sort of gives you a rationale for what you see, because it says the following, that if you are in a rich medium, in a medium that has a lot of sugar, then it makes sense to invest on growth, but growth requires proteins, and making proteins requires ribosomes. So this means that the richer the medium in which you have, the faster you can grow, the more ribosomes you need. While on the other hand, when you are in a poor medium, you need to invest in catabolic proteins because you need to be able to process the little sugar that there is in the medium efficiently. So this makes sort of intuitive sense. It's very appealing. However, how do you prove that the growth rate is actually maximized? Now, if you think about it, this is really hard. It's very hard. I mean, as intuitive and as easy to understand as it is that cells want to maximize their fitness, when you try to understand whether it's possible to actually prove it in the lab, it's a mess. Because how do you prove that something is maximized? It's really, really difficult, especially because there's no theory. So in physics, we are used to being able to thinking in terms of variational problems, so stuff is being maximized because we have a theory that allows to make predictions, and then we can check the predictions of the theory. Okay, but here there's no theory. There's nothing that links the fitness of a cell to, there's no microscopic theory at all. Okay, so it's really, really hard to understand how you can prove that the fitness is maximized. So people have tried different things. So one thing they tried comes again from a simple reasoning, which is the following. If you look at a cell, basically the mass of the cell is essentially the mass of its proteins. Okay, at least the dry mass of the cell. So once you remove water, it's essentially only proteins because everything else is extremely is negligible compared to the mass of the protein. At the same time, the growth rate is proportional to the rate at which you synthesize proteins, because in order to grow, you need proteins. And the protein synthesis rate is proportional to the ribosome activity. Again, this is intuitive. So if the growth rate is optimal, then this means that the ribosome activity has to be optimal. So we may be able to see that if we change the ribosome activity in the cell, somehow the growth rate decreases. Okay, this is something that has been tested again several times in the last maybe 20 years. This is one of the papers that I've done. This paper by Bollenbach et al. Has done a really nice experimental check of this. And what they did is basically, in E. coli is a bacterium, and the bacterium E. coli has 7 different places, 7 different loci on its DNA that correspond to the synthesis of ribosomes. So what they did was they simply removed selectively certain numbers of these loci so as to make ribosomes expressed less efficiently in E. coli. And what they found is that if the growth rate in the wild type, so in the bacterium in which all 7 loci are available, is up here, when you start removing loci, the growth rate gradually goes down like that. Okay, so you have to ignore the red line, just focus on the black. Okay, now I understand this is not much of a proof of optimality, but this is as far as you can go with the ribosomes. Again, it's hard to think about how to do something better than this. Because for example, it would be natural to say, okay, but I want to know what happens if I add one locus, okay? But then the result will depend on where on the DNA you add this locus. And you know, you cannot try all the places on a bacterial DNA experimentally to add the locus and see the effect that is that. So it's really complicated to come up with a good way of doing that.
Andrea, one question about this figure, if I may.
Yeah. Yeah.
So the last point, what is the last point in the black curve that goes because you're saying that
sorry.
Yeah.
So this is a point in which they have removed, they have canceled some, this is a mutant where they have removed some other promoters that produce some proteins that are, let's say, auxiliary to produce ribosomes.
Okay.
So this is, again, a way to reduce the ribosome activity. Sorry, I didn't explain because it's a little bit more complicated, but it's a similar thing. So you still go down in the growth rate. And the line is just something they added to make it easier to look at the plot. I don't know why. Okay. So another thing that you can expect has to be optimized if the growth rate is maximized is protein levels, right? So protein have to be expressed at the correct concentration in any given condition if the growth rate has to be optimized. And this is something that is a little bit easier to show. And this is, for example, shown here. So in this case, I think this is for E. coli again. This is for a different bug. It doesn't really matter. So here they took in an environment in which E. coli grows on this sugar here, it's called succinate. They just titrated the level of a certain enzyme. It's actually an important enzyme, an enzyme that is essential to synthesize ATP in cells. And titrated simply means they changed its level of expression in a biochemical way. And essentially the level 1 x equal to 1 here corresponds to what you have in the wild type, so in what is in the natural cell. And above here you have higher expression levels, and below here you have lower expression levels, and you see that basically the wild type gives you the fastest growth rate. So this kind of evidence you also have for other enzymes. And of course, it tells you that some enzymes or some proteins are actually regulated for growth, to optimize growth. But of course, you can't check all of the proteins for that. I mean, you just cannot do it. So this is what happens when you don't have a theory that way. Okay, however, this idea that that the growth rate is maximized was helpful in actually setting up a theoretical program that could be used to predict what cells would do. And again, for a physicist, this is relatively easy. So you imagine that you take a vector that encodes for the concentrations of all molecules that are inside the cells. And the concentration C is just the ratio between the number of molecules and the volume of the cell. Okay, and when you take the time derivative of C, you just end up with this, oops, sorry, which you can rewrite in this way. Now, if you are at balanced growth, these two terms are by definition identical, okay? Because at balanced growth, extensive quantities grow exponentially with the same rate. So this parenthesis here is actually equal to 0. So this means that at balanced growth, concentrations are stationary in time, which I said above, which I told you before. However, you can take one step back and look at this expression here, and you immediately realize that this quantity here is just the growth rate. So dV over dt times 1 over v is just the growth rate, times the concentration n over v. While this quantity here you can write as, gives you the idea of, let's say, the rate at which the number of molecules increases per time per volume. So the rate, the number of volume change per time per volume, okay? And you can just write this in terms of a matrix that encodes for all the processes that consume or produce molecules times a vector of rates at which these molecules are produced or consumed. And of course, the rates are just functions of the concentration. So at balanced growth, these two quantities have to be identical. But if you just look at metabolism, so at the processes that produce energy in cells, and you ignore protein synthesis, which occurs on much longer time scales, essentially what you can do is neglect this term compared to this, simply because in metabolism, this is much faster than this. And so you end up with a condition where this vector of rates V no longer depends on concentration. It's just a bunch of numbers. So if you know the reaction network, finding the reaction rates that are compatible with balanced growth is just a matter of solving a linear system of a homogeneous linear system of equations. Okay, so this is something that in principle is doable. So this is a nice theory, and people have done it. And I will not go into the details of how this is done, but there is a huge literature on this. This kind of theory is called flux balance analysis. And essentially starting from conditions of this type where the matrix S, the set of processes that form the matrix S are just given by the set of reaction networks that occur in cells, they are able to give you a prediction for what will be the maximum growth rate that is attainable by these cells, okay? And this is a really highly successful theory. It's widely used. It works very well for some things. Okay, but then, okay, one should see how good it is at predicting the actual growth rate of cells. Okay, you can predict some things, but how well does it actually do when you compare it with experiments? So this is a plot where we have plotted the predictions coming from flux balance analysis. This is just the red line for the growth rate of a bunch of E. coli cells on glucose, growing on glucose as a function of the glucose uptake rate. Okay, this is the x-axis, and this is what FBA would predict, and this is what you find in the lab. So as you see, this is not bad. Okay, this is more or less, this is not so bad, but it's clear that we're not quite there. So there's a lot of work that is being done in order to literally bring this black line down here. And the way people want to do it is by basically adding more constraints that will essentially make it harder for the cell to grow at any given glucose level. Now, to do that, they obviously include in the model a lot of biochemical constants, biochemical knowledge, regulatory knowledge, and then you have to get into the details of it, and then it's you know, it's complicated and detail dependence. And so what we try to look at is a different type of way of rationalizing empirical evidence. So one of the things that set us out here was essentially this term here. So this offset here that I was talking about. So the fact that it appears that even if the cell is in a medium in which it cannot grow, it still has some ribosomes left. Now, of course, cells have to survive, and they probably do need some ribosomes for that. But let's say it's worth asking what these ribosomes are doing during balanced growth. Are they doing anything at all during balanced growth? And as a matter of fact, it's relatively easy to show. So this is a model that is based on a paper that came out 10 years ago, that essentially you can explain this, this offset in a very simple way by just assuming that the ribosome pool in a cell is partitioned in 3 different classes. So there are some of the ribosomes are used to produce proteins, general proteins, so metabolic enzymes, you name it, RNA polymerases, anything, okay? Some other ribosomes are used to produce specifically ribosomal proteins, so they have basically to replicate, produce more ribosomes. And there is some ribosomes that instead are idle. They don't do anything at all. And if you study this model a little bit, it's a really, really simple linear model. You find that the ribosomal fraction phi actually has to depend on the growth rate in this way. And the offset that you see here corresponds exactly to the fraction of idle ribosomes. Now, this is intuitive, right? During balanced growth, they don't do anything at all. But then the question is, why are they there?
Yeah, sorry. Yeah, I was going to ask that. When you say idle, is because now we don't know what they are doing, or is?
Okay. So let me see if
Okay.
if I can answer this question now. Okay. The question is, why are they there? Because intuitively, if the cell got rid of these idle ribosomes, it could grow faster, right? This is what, because it could just free up resources that it could use to produce useful things, proteins that it actually uses, Okay. However, if this was the case, you would see that in long-term evolution experiments, they would get rid of this idle ribosome, but as a matter of fact, you don't see that. So cells keep these idle ribosomes throughout their growth, even in long-term evolution. Right? Okay, so people have to conclude that in order to maximize the growth rate, there is some mysterious reason why, even if you keep some totally idle ribosomes inside cells, this is convenient for growth. Now,
Andrea,
to answer,
sorry,
yeah.
Can I ask something? Could this thing be related to this time lag that you have at the beginning of the growth? So it is
okay. So
some idle ribosomes to start.
That's actually a good point. So what people have shown is that these idle ribosomes are useful when cells switch from an environment to another. So when they have to adapt ex novo to a new environment, they have to go through a lag phase. They usually do it more efficiently when they have some of these idle ribosomes. Of course, it's really hard to know if they're doing anything at all, if they have what they are doing. Okay, because you have to look at the level of the individual molecules. That is really hard. But okay. But anyway, if you focus on balanced growth, they seem to keep these idle ribosomes, even if during balanced growth, they are actually idle. And they're not the only idle proteins during balanced growth. People have shown experimentally that cells express a lot of proteins that they don't use, not only ribosomes during balanced growth. And if you manage to reduce their expression level, they could grow cells could actually grow faster. Okay, so this is really hard to explain. Why do cells express so many proteins they don't seem to need during balanced growth? Okay. Now, let me give you another piece of the puzzle, which is the following. I told you about these linear dependencies, and I told you that sort of they can be rationalized, let's say intuitively, if you think about protein synthesis rate in the case of ribosomes. But actually, this is much more general, and it sort of makes sense that all these fractions, they change linearly with the growth rate. So what you have to think is this: imagine a certain proteome sector, so a group of proteins that sort of perform the same function, and call mx its overall mass. And then it changes in time according to how many ribosomes there are in the cell, to how many, what is the fraction of ribosomes that is devoted to synthesizing proteins in the X sector, and to let's say a kinetic constant that tells you what is the mass of proteins that is produced per unit time per ribosome. So when you sum these conditions over the different proteome sectors, you find that the total cell mass changes in time in a way that is proportional to the number of ribosomes. Now, if you assume that you are in balanced growth, then all of these sector masses change exponentially in time. And if you plug these in here and then in here, you end up with this. This is really easy. Okay, and the only thing I've done is replacing the number of ribosomes with the ratio between the ribosome mass and the mass of the single ribosome. Okay, and from this condition, you can essentially reproduce all of these things. So the ribosomal fraction is actually very easy. You just divide everything by m, and what you see is that the mass of the ribosome divided by the overall proteome mass is just proportional to the growth rate. And of course, if you add the idle fraction, you go back exactly to this. Now, what is interesting about this derivation here is that there is no optimization. Okay, nothing is being optimized here. We are not assuming that the growth rate is maximized in any way. This is just a statistical law. This is what you should find, just because there are some physical constraints that tell you that the proteome fractions have to be proportional to mu, independently of whether mu is optimized or not. So there is no reason to think that mu is optimized based on this kind of behaviors that you observe. Okay, let me show you another piece of evidence. So this is getting slightly technical, but okay. So this is a kind of cartoon of the metabolic network of a cell. Imagine a bacterium. And you can distinguish two types of substrate usually. They are called A and B because, I mean, people didn't want to think better names. So basically, A substrates are those that are processed through this linear pathway here that's called glycolysis, essentially. While B substrates are processed through a different cyclic pathway that is called the TCA cycle. Okay. Now, of course, the way in which cells use type A and type B substrates is very different because the biochemistry here is really very different. In particular, it is known that category A substrates tend to be used. So when you place a bunch of cells in a culture in which there are different category A substrates, it usually uses them all together. So it doesn't make any distinction between different types of substrates unless you are in a culture, in a type of culture that is called batch. So in a dish, imagine. When you are on a dish, cells use them hierarchically. So hierarchically means they first use one type of sugar, and then when that is totally consumed, they use another one. And then when that is totally consumed, they pass to another one and so on. Okay. Now, the way in which the substrates are used in the hierarchy reflects more or less how fast cells can grow on that sugar. So they usually use the one that allows them to grow faster, but that's not always true. So the hierarchy is not perfect in this case. On the other hand, when you place cells in a dish in which there are both A and B substrates, they use them indifferently. So they don't make any distinction between the substrates. They use what they find. Okay. And the other piece that is important is that if you construct cell mutants that cannot make the hierarchy, okay, because they lack the specific enzymes that allow them to use things hierarchically, you don't see a change in the growth rate. Okay, so the hierarchy doesn't seem to be so important for growth. Now, if you think about what would be the optimal case, in the optimal case, you have to expect only hierarchy. And the reason is actually simple. So the growth rate has to be proportional to the, imagine that you have a bunch of bacteria in a culture in which you have two different types of sugars, right? So the growth rate has to be proportional to the proteome fractions devoted to one sugar and to the other sugar. And then if you optimize, what you only need to know is whether this constant or this constant is bigger. So for example, if this constant is smaller than this one, then the optimum is obtained when you just set phi1 equal to 0 and you just use phi2. Mm-hmm. So in other words, the optimum leads to a hierarchy, okay? So cases in which there is no hierarchy cannot be explained by an optimum. And there is something even more tricky here, that experiments in which they have grown cells in A plus B media, so in which you have both types of compounds, can actually grow faster than what the hierarchy would predict. So they found that cells have found a way to use the two substrates more efficiently than what an optimal theory would predict. Okay.
So Andrea, but that's I find it this very very counterintuitive, right?
Yeah. So the reason is that basically when you think about optimizing this, right, you optimize over phi1 and phi2 as if they are independent, as if you can change them independently. Okay, but
sorry, let me tell you why I think biological is counterintuitive. Because when you have substrates which are of both categories, say A and B, so I mean, chemical reactions cannot do more. So for instance, if the tricarboxylic cycle is active, okay, because you have substances in category B, so nothing that comes from glycolysis will help you to boost the Krebs cycle, right?
Nothing that comes.
I see. So you see what I mean, or no?
Well, I mean, all of these are sort of reversible. So you can always go upstream in glycolysis and then come back here. So in principle, if I understand you correctly, in principle, there is actually a positive feedback possible through a pathway that is here, essentially, which is called the pentose phosphate pathway.
Okay.
So in principle, it's not like, let's say, generally speaking, category B substrates are less efficient to grow on, so you grow slower, because the processing through the TCA cycle is a lot more costly in terms of proteins than processing through glycolysis. Glycolysis is very easy, very linear, and there's only a few enzymes here, while the TCA cycle is a huge mess, and you really need to invest a lot of molecular resources into running a TCA cycle. So this is the reason that most people think that category B substrates are less efficient.
I see. Okay.
But in principle, you can have a positive interaction between the two. And actually, this is what is going on here, because when you optimize, you imagine that you can change phi1 and phi2 independently, but if they are mutually dependent, then the things are not so easy, right? So it can be that you actually grow faster than you would predict by a straightforward optimality theory. So, oops, what's going on? Okay, so to summarize, there are good reasons to think that cells maximize their growth rate, but there are also good reasons to think that maybe, you know, there are some things to understand that are not totally, do not totally agree with the optimality idea. So our approach here was, let's say, a physics approach. And we started by going basically back to the first slide or the second slide I'll show you, and to start to look at what cell-to-cell variability means in the context of balanced growth. So let me be clear. I told you that cells in a cell population are heterogeneous. They're not all the same, but in balanced growth, some quantities obey stationary probability distribution that can be measured in the lab. And the way you measure them is by just taking, for example, in specific microfluidic apparati, you can take movies of the cells growing, and from these movies, you can measure different things. For example, the size at birth, the size at division, the interdivision time, and so on and so forth. It's just a relatively straightforward, but quite informative technique. From this technique, you get these kind of distributions. And there is something that is really striking about these distributions, namely the fact that there are some cells. So if you look at, for example, this distribution here, there are some cells that have very long generation times. So they're very slow at dividing, but there are some cells that are really, really fast at dividing. They're down here. So the question is, why don't these cells take up the entire culture, right? We said that selection should work, because if there is a cell that is very fast at dividing, then you know, its sons will eventually take over the entire culture simply because they are faster at eating all the glucose there is. But this is not what is going on. Okay, this seems to be not what you find in experiments. And also another interesting thing here is that basically, if you just by looking by eye at these distributions, you see that essentially as the mean increases, of the mean of these distributions increases, so these correspond to different growth media, also the variability increases. So the sigma of these distributions, if you crunch the numbers, is actually proportional to the mean. So if you assume that the mean of these distributions, which is a very good proxy for the specific growth rate, is optimized, then you also have to assume that the variance is optimized. So, but then you have the problem of understanding whether it's the mean or the variance that is being optimized. So do the cells attempt to grow faster and faster, or do they attempt to make their behavior as heterogeneous as possible, so as to be able to face conditions as different as possible? Okay. So as physicists, we of course thought of these distributions as samples from a phase space that was that represents the state of, for example, the levels of proteins that are accessible to cells. And when you observe this data, this just means that you're sampling different parts of this space. So what we did was something, conceptually simple for physics. Namely, we thought of associating to these distributions two quantities, namely its mean values, their mean values, which corresponds to their fitness, so their specific growth rate, and their entropy, so a measure of how spread they are over the feasible space. Okay, I'm not going into the details, but essentially when you take details are rather technical, but you can essentially take experimental data for populations growing on a certain medium, reconstruct the probability distribution that corresponds to that particular experiments, and from that extract fitness and entropy values. And then you can place them in a plot like this, where there is fitness on the y-axis and basically minus the entropy, so what is called information, on the x-axis. And each of these points corresponds to a different experimental population that is actually from the blue to the red is grown in better and better sugar conditions. So they could grow faster and faster. And what you see is that, as a matter of fact, their fitness increases and the information content in the distribution also increases. Okay, so this means that as they get faster and faster, also the distributions get less and less disordered, which is what the data were telling you. However, once you have this mathematical structure, you can also have a prediction for an optimal performance. Namely, you can calculate what is the maximum growth rate that you can achieve at any given level of information encoded in this distribution. So you can say how far from optimality these distributions really are. Now notice this is very different from before. In the previous case, the optimal state was point-like, let's say it was a single state. Well, in this case, we are talking about optimality for distributions. So there's always heterogeneity here included. And what we find...
Sorry, there is a question in the chat, sorry to interrupt, that says, Mario is asking, why is there a breakpoint at 0.48? I guess the
okay, so the breakpoint is due to the fact that these experimental data points were taken from two different papers. So the blue ones are taken from a paper by Nanchen et al, and the orange ones are taken from a paper by Schutz et al. It's the same group. They just divided their experimental data set in two papers, one for slow growth rates and the other for fast growth rates. This is what they did. So the protocol is exactly the same. So the whole experimental setup is the same across the two papers. It's only that they wrote it up in two different papers. So it's two different files, dataset files. And we wanted to keep the distinction here. Okay, that's the only reason.
Thank you.
Okay. So what you see here is that basically as the growth medium gets richer, so as you move from blue to orange, the points actually do approach optimality, but they stay suboptimal. Okay, so growth does not appear to be optimal, even if you include fluctuations. Okay, now this is not let's say a qualitative statement. This is a quantitative statement. So this is based on these probability distributions that we can actually reconstruct and match against the data. So what this suggests, but anyway, the nice thing is that as you get faster and faster, you approach optimality. And if you take populations that are growing really, really fast, they are not included in these papers, they are from a different database, you can actually see that the probability distributions of the growth rates, single cell growth rates that we predict, actually match experimental data really, really well. So again, these are populations of bacteria that are strains, modified strains that are growing very, very fast. So according to this picture, they should have approached the optimal behavior. And so indeed, the optimal probability distribution, which is the continuous lines here, gives a very good description of the data points. Okay. So I will skip this in the interest of time, and this brings me to a conclusion. What I hope to have shown is that we are at a point where I think it is possible to make sense of a very, very difficult question in biology, of a very difficult concept in biology, which is that growing cells maximize their fitness. Okay, this is something that we are really used to think of, and this is something that in many cases makes a lot of sense to believe, and this is something that in some sense it is reflected in data. But when you think about it, it's really, really hard to come up with a falsifiable test, a falsifiable theory that is based on this idea of optimality. So it's very hard to validate it experimentally. Also, there is the question of what is being optimized. Sometimes it seems that the growth rate is being optimized, but then you have to make sense of the idle ribosomes, for example. Then you have to say, okay, then maybe it's also optimizing how fast it will adapt to the future environment. So it is sensitive not only to its current environment, but also to what it can possibly happen in the future. Okay, and then the other problem of optimality is that even if you're very good and can do flux balance analysis is a theory that has a lot of parameters. So, I mean, making predictions when you have so many parameters to set is not, let's say, it's not ideal. On the other hand, the kind of approach that has been developed by us, but not only by us actually, which I would call a statistical approach, is based on the empirical evidence. So it's based on the data, and it tries to reconstruct cellular behavior from the data rather than postulating it somehow. So it accounts for cell-to-cell heterogeneity. So it's not something that focuses only on one point in the phase space, only on the optimal point, but it deals with heterogeneity as part of optimality. So optimal behavior is not only in terms of the mean fitness, but also in terms of fluctuations. Of course, the constraints are essential, so we need a way to define the phase space, and this is a constraint-based problem. So we have a problem with the constraints we don't know about. So this can be a very reasonable criticism, in my opinion. But on the good side, it generates a lot of testable predictions that can actually validate it in the lab. So with this kind of approach, you can push it to the point of having predictions for growth laws that have not yet been found in the lab. And I didn't have time to talk about this, but I mean, this is something that is worth doing. So let's say at the interface between physics and biology, I think there is a really interesting, let's say, discussion that is going on that revolves around the question of what actually is the fitness of an organism. Does it coincide with the growth rate? And if so, what does it mean that it's being optimized? Is it being optimized? And if it's not being optimized, what are these cells actually doing? So I think this is an interesting time to look at this type of problems. And interestingly enough, biologists have been looking at these problems in the 50s and 60s, and then they forgot about it because the whole era of genetics started, and that of course became a much bigger topic. But if you're interested in a historical perspective from the biological point of view, I have this recent review to suggest it's not, maybe it's not the kind of article that a physicist will read, but I found it really inspiring to show that actually some questions are really, really deep. And even if they don't come from physics, in the end, it might be that physics has the keys to answer some of these really interesting questions. So sorry for being maybe a little bit too long, and thank you very much for listening.
No, Andrea, thank you very much for this. I mean, fantastic talk. I mean, I told you before in another seminar you gave the workshop in the Max Planck Institute that I think the work you do is very inspiring. I find it extremely interesting. So if colleagues have a question, so please go ahead, just unmute the microphones and ask directly to Andrea. Or if you have some technical reason, maybe write it on the chat and I'll gladly read it. So I think Mario maybe has a question. Please, Mario, go ahead.
Yeah, thank you very much, Andrea, for this really nice talk. So probably I missed it, but I wonder which is the technique that you're using to measure this kind of rate of changes. So it's a particular technique to obtain the proteins, or how do you measure the rates of changes?
So let me see if I. So you're referring to these variables here?
No, I mean, so on the whole system, how you define or how do you measure the actual change of rate between your protein consumption with your number of cells?
So you mean sorry, Mario, you mean experimentally rather
Yes,
than
correct.
okay.
Experimentally. So okay, that's a very good question. So this is not actually the job we have done. Because we have been dealing, let's say, with this part of the job. So we took the data and we processed the data in a way different from the usual. But I can tell you how these fluxes are actually measured. So there is basically the technique is mass spectrometry. Sorry, could I just ask to mute the microphone because, sorry. Yeah, thanks. Because okay, so the technique is mass spectrometry. And the way they do it is they feed the bacteria with glucose that has in one specific position in the glucose hexagon of carbons, they have replaced one C12 with a C13. And so they can see the signal coming from this C13 in the mass spectrometry, and they can see how the C13 distributes over the downstream product of metabolism. And from that, they can reconstruct how much C13 went through the different metabolic pathways. And from there, they infer the fluxes. This is how they do. So of course, being a flux, it's a dynamical variable, so you can only measure it indirectly, if you like. But the technique is actually quite well established, and this is the one that was used in these two papers to quantify the fluxes, and the one that we used for the distributions. On the other hand, there is an assumption, I mean, a key assumption that I sort of said at some point, maybe I should have stressed it more, is that when you use this kind of representation that is based on this equation, you are assuming that you're looking at what cells do over time scales that are shorter than those required to synthesize proteins. Okay, so because otherwise, basically you cannot neglect this term compared to this. And then things become a lot harder. So let's say a background assumption of all this theory is that contrary to the metabolite levels, which change given by those rates, protein levels and macromolecular levels don't change. So this is the other bit I think that is required to answer your question. I hope I have.
Yeah, indeed. I was wondering which was the technique that you use, because as far as I know, at least related on my field, in MRI or with the spectroscopy, there are some kind of techniques where you can measure this kind of switching pulse that you were talking about in the I think one of the latest slides, where you can measure this flux balance, and actual you can measure it with the spectroscopy line shape of the cell. So that's why I was wondering.
Yeah, that's absolutely correct. Yes, yes.
Thank you very much.
Thank you, Mario. Mario, more questions? So if you have, please unmute your microphone and ask directly. Or write on the, you can write on the chat. Andrea, I have a question myself. So from okay, we have been told, okay, as you were mentioning at the beginning, that this issue of optimality is maybe it's due to some kind of evolutionary reason, right? So cells want to grow very fast because due to natural selection, okay, since the appearance of the first simple cells 4 billion years ago, etc., okay. So it appears that this is not actually true, right? So from the point of view of evolution, what is that is being optimized in a cell? It seems like it is the readiness of the cell to change rapidly and to accommodate to environments, right? Rather than that grow very fast.
Yeah. Okay. This is a very good question. So so basically, from this kind of plot, what you can say is that at least let's say when the quality of the medium in the environment improves so that cells can grow faster and faster, they actually do try to maximize their fitness. However, the cell-to-cell heterogeneity takes into account the fact that the environment is not a delta function; it's variable.
Mm-hmm.
Okay, so in this sense, I think the most the most reasonable thing I feel like saying is that really fitness is a population level concept. So well, the population tries to adapt, tries to find a state in which this kind of variability that you observe here matches the sort of variable conditions that they have to face in the environment where they are.
Yeah, I guess that somehow you're optimizing, the cell is optimizing its capacity to store information about the environment and how this information can be used.
Yeah, for example, keep in mind that we are always thinking about balanced growth. So the ingredient of switching environments isn't really present here. So from this kind of experiments, we can't really say what happens to cells that face very, very variable environments.
I see.
Okay, so in this case, we are in a relatively stable growth conditions, but still the environment, let's say, implies sufficient variability, sufficient fluctuations, maybe not so much in the environmental variables, but in the intracellular variables that talk to the environment. So in the protein levels that are required to deal with that environment, they can be sufficiently variable that a large variability is required. Okay. Also, I think that from an evolutionary viewpoint, it makes a lot of sense to think that cells want to keep variability, because this is again a sort of anecdote that you may have heard of, but essentially, when you subject a population of cells to some sort of antibiotics, so bacteria to kill them, for example, you can usually, it's very easy to kill the cells that grow fast, so that the ones that are in the left tail of this plot, but usually you don't kill the slow growing cells, because basically they are not growing because they are, let's say, reserving a part of the energy to synthesize macromolecules that will help them in case they meet antibiotics.
I see.
Right.
That's
Very good.
Yeah.
So at a population level, the idea of keeping a large variability seems to be sensible evolution from an evolutionary viewpoint. But I think that this is quite a jump from a conceptual viewpoint, because it then says that really the fitness is a population level concept. So when we think about regulatory mechanisms that act at the level of a single cell, we should always keep in mind that there is a level above that, which is a population level, where things are organized in such a way that all these regulatory mechanisms in the end must allow for some cells to be in this tail here, because it's useful for the population. So it's really So the more you think about it, the more it's like Inception, the movie, you know.
Yeah, that's right. Yeah, that's right. Yeah, very good. Okay, one more question from the colleagues in the audience. If not, so Andrea, thank you very much. Fantastic talk.
Thank you so much. Wait, I'll turn on the light, otherwise.
Yeah, because
I'm in the dark.
yeah, your background became darker and darker.
Yeah, I'm a creature of the night, so I like living in the dark.
Yeah, thank you very much for this fantastic talk, Andrea.
Thank you for inviting me.
Here. Yeah, of course, a pleasure. I hope, I didn't mention this thing, but okay, I should have said welcome virtually to Mexico City. You were here once, I don't know, maybe 3-4 years ago, because you were passing by going to the States. So yeah, let's hope that in the near future you can come back to visit us. Okay.
Totally.
Take care, Andrea. Thank you,
guys. I will speak Spanish.
I hope you will. Yes.
Yeah.
Take care. Bye bye. Gracias a todos. Nos vemos la semana que viene.
Chao, Isa.